Hey everybody, how's it going? Hope everyone's doing well. Hope everyone's staying healthy and staying the heck away from everybody else and washing your hands a truly ridiculous number of times. So we're going to kind of get started up on our lecture now that we're past spring break and extra spring break and extra spring break. Okay, cool. We get the point. Today I'm going to be lecturing on chapter 14, which is DNA. And I know we've talked about DNA before, but we're going to talk about DNA again. We're going to talk about how we figured it out. We're going to talk about a lot of things: the structure, how it can screw up, how it replicates. So that's what this lecture is going to be about. And yes, there is absolutely a reflection on my glasses of my monitor, and I know it's annoying as heck, but luckily you don't have to look at me much longer. And the only thing we can do about that is this, and we don't want that, do we? Yeah, thanks. So let's see if I can get this thing switched over to the correct place and settings. And I guess I should have been in the right place and been planning for this, right? Okay, so DNA, we're going to talk about DNA. Excuse me. You should absolutely recognize that molecule right there, that polymer, that is DNA. So we know that DNA is a genetic material, and we're going to talk a little bit about how we figured it out, right? We have already talked about Mendel and his work with sweet peas and whether the color of the flower was purple or white, or whether the seeds were wrinkled or smooth, right? And those are Mendelian traits. And whether, which trait was dominant, which was recessive. We're kind of past that, right? So we're going to move on now. We're going to talk about Frederick Griffith. Now, Griffith, this is back in 1928. He was working with a bacterium, Streptococcus pneumoniae. And Streptococcus pneumoniae is pathogenic. Gotta love that word. You probably heard that a lot lately. Maybe, maybe not, depending on how much you're following this coronavirus outbreak worldwide, pandemic. Yeah, lots of fun. So, pathogenic meaning, it means it's disease-causing. And so, he had one strain that was absolutely disease-causing, Streptococcus pneumoniae. Well, he had two strains. One was virulent, and virulent means it will make you sick, and non-virulent means it will not make you sick. So he called them the S strain and the R strain. The reason he called them that is the S strain, when grown in a petri dish on media, the edges of the colony is smooth, and the R strain, the edges of the colony are rough, so smooth and rough. What causes the smoothness in the S strain is that it's got a capsule around it, and it's that capsule that helps it to evade our immune system, and so it helps it to be much more pathogenic. So Griffith, like any good biological scientist with virulent bacterium, injected them into mice. He infected mice with these strains, trying to figure out what was going on. And so when he injected live S strain, remember the virulent strain, he thought it should probably kill the mice. And indeed, it did kill the mice, just like he thought. Then he took live R strain, which is avirulent or non-virulent, and he injected it into other not dead mice, and it did not kill the mice. Hey, just as he thought it was supposed to happen, right? Then he took some of the virulent S strain and he killed it, basically with a tube over a Bunsen burner. He heated it up till all of the bacteria were dead. And so he thought that's probably not going to kill the mice because they're dead. He injected the heat-killed S strain into them and didn't kill the mice. We're still on point. He's getting exactly what he's expecting. But then he takes some heat-killed S strain and some live R strain, mixes them together and injects that into the mice. Now, the S strain is dead. The R strain is live but avirulent. And yet, this mixture killed the mice, and that freaked him smooth out. He thought that was pretty strange. That was not what he really expected, right? So, here we have it. Live virulent S strain is injected in the mice. The mouse dies. Live non-virulent, the mouse lives, right? Next one, heat-killed virulent strain. So we got the S strain, which is virulent and deadly, but has been killed. Injected in the mouse, it's dead. It doesn't bother the mouse at all. Great. So then he takes a mixture of the aforementioned heat-killed virulent strain and some live non-virulent strain, injects it in the mice, and lo and behold, the mice die. And that blows his mind. Yeah, we're not to Hershey and Chase yet. So what's going on here? What he figured that was going on was that there was some particle, some thing that was in the dead virulent strain that was being transferred to the live non-virulent strain to make it virulent. And in fact, there was. And we call this transformation in biotechnology because, yes, we can't have a lecture where I don't talk about biotechnology. In biotechnology, we will move genes from, say, one bacterium, one strain of bacteria, or from a human, or from whatever, to a bacterium like E. coli, which is our workforce. And what we call that is the same thing he called this is transformation. We are transforming, in this case, the E. coli that we're using for biotechnology, or in the case of this experiment, we are transforming the non-virulent strain of Escherichia into a virulent strain. But he didn't know what that stuff was, but he knew he had found stuff, right? So then we get to Hershey and Chase. And Hershey and Chase did some really, truly groundbreaking investigations, and they were doing it with something called bacteriophages. Now, I hope you remember that one of my favorite critters is a bacteriophage, and I hope you remember that just because it's got bacterio in its name, it is not a bacteria. It's a virus. Bacterio means, well, bacteria, but phage means to eat. So bacteriophage, to eat phages. So these are viruses that infect bacteria. It's not really a food thing, but it uses the bacteria to make more viruses, right? So that's what's going on. So bacteriophages are very cool. So viruses infect bacteria. And these bacteriophages that they were using, and most bacteriophages are formed like this, and yes, they are the ones that look like lunar landers. Bacteriophages are very simple things. We don't even necessarily think they're alive qua alive. But a bacteriophage in this case are made only of DNA and protein. They have an outer protein, well, called a capsule. We call it a capsid or coat. And that's on the outside. And on the inside is what it contains is the DNA. So these viruses are made up only of an outer protein capsid and with the DNA inside. And they wanted to determine which of these is actually going into the bacteria, because whatever's going into the bacteria is what's doing the thing, is what's making the thing happen that makes bacteria start making viruses rather than more bacteria. Okay? So they actually did this in a very cool way. They did it with radioactive stuff, right? So we're going to be later going over the structure of DNA again. I know we've been over it, but we're going to do it again. DNA contains phosphorus. Very important here, protein does not contain phosphorus, okay? Protein contains sulfur, but DNA does not contain sulfur, and that's what they were using. They were using this fundamental difference between these two types of molecules. So they grew one bunch of bacteriophages in a media that had phosphorus 32 in it. Phosphorus 32 is a radioactive form of phosphorus. And so as these new viruses are being made in this media, the DNA is incorporating, at least partially, this phosphorus 32 radioactive isotope of phosphorus. And of course, radioactivity is traceable. That's why they were doing it. Another bunch of bacteriophages were grown in a media that had radioactive sulfur in it, sulfur 35, okay? And again, as they are growing and new virions are being made, those new viruses, the protein, whatever is made of protein in them, incorporates this radioactive sulfur 35. So now we have a strain, if you will, of bacteriophages with DNA that has radioactive phosphorus. We have another strain of the same bacteriophage, but another strain that has sulfur 35, radioactive sulfur, in its protein. Now we can get some work done, right? So let's see, we should have here we go. On the top, they first let the phage grow in radioactive sulfur, which remember is in the protein. They allowed them to infect bacteria, and they did. And then they beat them all up and broke up all those bacteria. They spun everything down. And what happens when you're spinning down, the heavier stuff goes to the bottom. So all of the solid stuff, if you will, which includes all of the contents of the cells, the interior of the cells, is made into a pellet, forms a pellet right down here. And the rest of this stuff we call the supernatant, and that's the other liquidy lighter stuff that's left over. So anything that was solid and had been in the pellet it was in the bacteria, right? So when they did this with the sulfur, which means it's in the protein, the radioactive stuff was up here in the supernatant, was not in the pellet, which means it was not in the bacteria. So they took the other strain that's got phosphorus 32, and remember, DNA has phosphorus. So it infected the bacteria, they broke it all up, and they spun it all down, and they found all of the radioactive phosphorus down in the pellet. And I shouldn't have hit that key. Down here in the pellet. And what that meant was they now knew through this experiment, which was repeated, of course, by their peers many times, we now have really good evidence that DNA is the stuff that's carrying information or hereditary information. This was a huge deal. It's a big deal. So, and you want to know about both of those experiments. I have a video already listed about the Griffiths experiment. I have another one that covers both the Griffiths and the Hershey-Chase experiment. Watch them. They will make much more sense when you watch them. So just DNA structure. Well, let's skip the writing and just go straight to pictures, right? So this is a nucleotide. This is a single nucleotide. All nucleotides are built this way. All nucleotides have exactly this part. The phosphate group and the pentose sugar. In this case, because it's DNA, it's deoxyribose is the pentose sugar. And they're connected here, and they form what we call the backbone, which I will get back to. This is the part of every nucleotide that can be different. This can be an A, a T, a G, or a C. So this is the actual nitrogenous base. So we have the purines, which are, of course, this large structure, double ring structure, and they are adenine and guanine. And then we have these smaller ones called pyrimidines that are single ring structures, right? And they are cytosine and thymine. We're not going to worry about uracil right now because we're not talking about RNA. We're talking about DNA, so we'll be talking thymine. All right. So if I ask you a question about purines and show you a picture, for instance, and it's a double ring structure, just remember that the larger structure has the smaller name, and that will take care of you going the other way too, where the smaller structure has the larger name. So again, this is the variable portion of all nucleotides, and this, in the case of DNA, can be an adenine, a guanine, a thymine, or a cytosine, right? This is a stylized whole nucleotide. We have the phosphorus group, we have the pentose sugar, we have the nitrogenous base. That's one nucleotide, and it is connected to another nucleotide by a phosphodiester bond. And no, you don't have to remember the structure. You have to remember phosphodiester bond, right? So if I ask you what holds nucleotides together in the backbone, you're going to say, you're going to choose phosphodiester bond, right? Okay, so Chargraff's rule. Chargraff was a scientist who was a slightly older contemporary of Watson and Crick. We'll get back to them. I'm going to put a link for a movie that I think you will all hopefully enjoy about the scientists who were trying to elucidate the structure of DNA, what it looks like, how it's put together. And Chargraff was a part of this, and he was really mean to the two guys who figured out what it shaped like, but it was really funny. So Erwin Chargraff, what he determined, that if you're studying a chunk of DNA, any given chunk of DNA, and you have X amount of adenine, you have exactly that same amount of thymine always. If you have X amount of cytosine, you have exactly that amount of guanine. And that's because I know you remember this, right? Adenine binds only with thymine and cytosine only binds with guanine, and vice versa, right? So that's why there's always the same amount of A to T and the same amount of C to G, because they selectively only bind with each other. That's Chargraff's rule. Okay, I told you I'd come back to Watson and Crick. Watson and Crick are the two scientists who are credited with having elucidated or figured out what the structure or the shape of DNA is. And they did. And I will say that Watson and Crick did exactly zero experimentation themselves. I'm not saying they were evil. I'm saying they weren't necessarily effusive about giving the correct people the credit they deserved. And who was that correct person? It's Rosalind Franklin. Rosalind Franklin was a researcher basically in the same institute that they were at in England. And she was an X-ray crystallographer. And she made these wonderful pictures, and it was very difficult. She would have to first crystallize whatever she was going to be taking pictures of, and then she would beam X-rays at that crystal, and they would refract the X-rays, and they would make these photographic images called an X-ray crystallograph, right? And this shape, this X shape, indicates pretty distinctly that whatever this is helical or spiral in shape, and it is in fact DNA, all right? And Watson and Crick were shown her work without her knowledge, and so they took that and ran, and they published very, very quickly, and they just happened to be correct, okay? Let me see. Here they are. This is the model they came up with the two backbones on the outside and the nucleotides stretching across to reach each other on the inside. And if you linearize DNA, it looks like a ladder, right? And that ladder is the two uprights that you put your hands on if you're climbing the ladder. Well, that I would put my hands on if I was climbing on a ladder. Those are what we call the backbone, and those are the phosphorus groups attached to the pentose sugars. The rungs that you stand on are the two nucleotides reaching across and meeting in the center. And so they proposed that double helix, and they almost barely mention Rosalind Franklin, but they went on in just a few years after publishing to win the Nobel Prize, which Rosalind Franklin could not share in because she was way dead. And she was way dead because she worked with X-rays at a time when, they still weren't too sure how dangerous that was. She didn't take a lot of precautions, and she ended up with ovarian cancer and died at a very young age, died in her 30s. And these guys got incredibly famous. One of them's still alive. I think it's Watson. And all I will say is he is well known. He is well respected, both of them. And he's well known as, well, being a jerk in person. We'll just leave it at that. Do your own research. Find out about this stuff. So it's a double helix. That just means there's two backbones that are winding like a spiral staircase down. It's a double helix. You've seen it and you will see more. I will be putting up videos actually at the same time or before I put this one up so that you can see these visually in video form. I think it will really help you to understand it. This is a double helix. We have two different strands, and each of them at each location has a nucleus reaching out to the center. So these are the uprights of a ladder. These are the rungs of a ladder if you were to unspin it, unwind it, and linearize it. Okay, what do I want you to know about this bonding of the A's and T's and C's and G's? Well, first, you need to remember that C's only bind with G's and A's only bind with T's. Big deal. A's and T's bind with each other with two hydrogen bonds. C's and G's bind together with 3 hydrogen bonds. So the CG bonding is 1/3 stronger, roughly, than the AT binding. And what does that mean? Well, when we do PCR, polymerase chain reaction, one of the things we have to do is break these hydrogen bonds. So when we sequence something so that we know what the sequence of A's and T's and C's and G's is, one of the statistics we really want to know is we want to know what's called the CG content. So say a chunk of DNA that we're studying, we just sequenced it, and we know statistically that it's about 50% CG. Well, okay. Then we can do PCR pretty normally. We can set it at, say, 60 degrees Celsius, actually higher than that, I think it's more 90, to break apart those hydrogen bonds and linearize them so that we can do PCR. But if we have 65% or 75% CG content, because CGs have that extra hydrogen bond, we actually have to add more heat to our polymerase chain reaction to make those two strands denature, to make all of these hydrogen bonds break. Replication. Okay. I'm just going to go down. We have conservative model, a semi-conservative model, and a dispersive model. And what this is telling you is not that these are the three ways DNA replicates. It's not. Only one of them is correct, and we're about to get there. So we're going to go to visuals. This would be conservative. This on the top is our parent structure, our parent DNA strand, and after replication, we still have our double-stranded parent. Both strands are parental strands, and the new strand is completely new. And that's not how it works. This is a lie, just like the cake. Okay, so we have that again. This is conservative. This is semi-conservative. We have the parental double strand, and after replication, semi-conservative replication would say that each of the new double-stranded DNA each has one parental strand, and ding, 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 ding. That's how it happens. That's actually how it happens. The third one is dispersive, and it's just nutty. So basically, this one ain't the way it happens, and this one ain't the way it happens. It happens this way. All right? Semi-conservative is what you want to remember. We're not doing Messelson install right now. We are in replication. And so you got to have three things to be able to make new DNA, to be able to replicate DNA. You got to have something to copy, so you got to have DNA. You got to have something to do the copying. You got to have enzymes, enzyme complexes, things like DNA polymerase, helicase, RNA primase. We'll get back to those. And we have to have the building blocks, and those building blocks are nucleotides, what they call nucleotide triphosphates. And those are just free-floating A's and T's and G's and C's that aren't connected to anything else, so that they can be used as building blocks to build the new DNA. All right? So there are three really easy to remember steps of DNA replication. They are initiation, elongation, and termination. Initiation, woohoo, that means it starts. It begins, right? Elongation means it's actively adding new DNA nucleotides to make new strands. It's elongation. Termination, I really hope you understand that this is self-explanatory too. Termination means it's done, it's stopped, it's over. That's when it ends. Those are the three steps, and you do want to know them. We're going to skip that one. Do I want this one? Don't want this one right now. I want to talk not about prokaryotes right now. I want to talk about eukaryotes. We're going to use some of this, but we're going to be talking about eukaryotes rather than prokaryotes. Let me go forward and see if it does this all over again. Eukaryotic. Yeah. No, they're not giving me the information I want. So I'm going to go back. Sorry about this. Okay. So we are talking about eukaryotes. Okay. I don't care that you memorize, because these are in prokaryotes anyway. What I want you to remember is not DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. I want you to remember DNA polymerase. Just DNA polymerase, okay? And I want you to remember helicase. Let's see. So DNA polymerase, let's go down to a visual. Here we go. Is that helping us? It's not helping us. I'm all over the place today. I haven't done this before. Here we go. So DNA polymerase is what actually adds new DNA nucleotides to the strands. First, I want you to remember helicase. Helicase is an enzyme complex that jumps in here and splits this DNA apart into two single strands. And this, right here, you want to remember, it's called a replication fork. Yeah, see, right there, replication fork. And that's where the helicase has landed and opened it up, okay? Then we have some other enzymes at work here. We have DNA polymerase, which is we're just going to take this whole thing as DNA polymerase working on both strands, right? So, you see, this looks like it's going really easy, right? Cool. What happens, DNA polymerase can only go chemically one direction. Okay? Can only go 5' to 3', this direction. And that's okay. This one's going the right direction. So what it does, and it's not being shown here, but out here, right at the beginning, one of these RNA primers has been put in place. And that RNA primer allows DNA primase to attach to it and begin doing its thing. Once it's found it, since it's going the right direction, it can just keep going to the end of the DNA. And this strand, what we call the leading strand, is very easy. It just needs one RNA primer, and it does its thing. Great. But remember, DNA is anti-parallel. This one up here is 3' to 5', so it can replicate 5' to 3'. But this one is 5' to 3', which means the replication is going to be going 3' to 5'. And it can't do that direction. So what happens is something called RNA primase is going to come in here, and it's going to lay down a primer. And then DNA polymerase can lay down here and put on all these blue, keep hitting the key, can hit all and put on all these blue DNA nucleotides. But then it comes to the end and it stops. Then RNA primase puts down another one back here, and DNA polymerase can come in here and fill that in. And then another one, and then DNA polymerase can fill in more. And this is called discontinuous replication. This up here from the leading strand is continuous replication. You need to know these two. The leading strand is made continuously. The lagging strand, which is what we call this one, is made discontinuously in chunks. And you see those chunks, primer, then DNA nucleotides, then primer and DNA nucleotides. That's the lagging strand. We call those chunks, those fragments, Okazaki fragments. Okay? You want to know that too. And yes, I will be having definite videos uploaded for you, video links for this stuff, because it's so much easier to understand when you watch it. So I need you to know helicase. I need you to know primase, or what I call RNA primase, because it puts down RNA primers. DNA gyrase. Again, it's hard to do this non-visually, but if you have two strands of DNA and you take one of each of the two strands in one hand and pull them apart, it's going to start supercoiling the rest of it. And DNA gyrase keeps that from happening. It actually continually cuts and glues it together. But visually, I'll try to find a video for that too. And you need to know DNA polymerase. And then if we go back up here, once all of this is laid down, and once all of this is laid down, this is almost done. There's only one more step. A couple more steps. We have to have another kind of DNA polymerase come in and remove these primers and put in DNA nucleotides. And then the last step for both of these strands is DNA ligase. DNA ligase glues all of these new nucleotides together on both strands. Glues them all together, make sure they're all connected and everything's nice. And once the DNA ligase has done its thing, we're done. Okay? And that, we were actually talking about eukaryotic replication. So we're going to go past some of this. We're not going to worry about telomeres right now. DNA repair. Well, first off, why would DNA need to be repaired? What happens? Well, DNA during replication can get screwed up. It can do it incorrectly. I think they say something like 1 in every 10 million replications has a single nucleotide screw up or a mutation. So it puts in the wrong nucleotide. Well, DNA polymerases that are doing this, they come back and they can do what they call proofreading. And if they see that, for instance, they got a T connected to a G, which is wrong because it could be to an A, they can excise out or cut out that T and put in a C, and so it'll work, right? A C and a G bind together. So what are mutagens? Mutagens are things that cause above normal happenstance of these screw-ups happening. So mutagens can be radiation, so, don't go getting bit by a radioactive spider. It will not turn you into Spider-Man. If you're lucky, it'll just give you whatever the spider's venom does, which is going to be bad enough in and of itself. Don't go eating, things that are emitting gamma radiation. You will not become the Incredible Hulk. You will just die. And, well, it won't suck anymore because you'll be dead, but it'll suck as it's happening, right? So DNA repair repairs these mutations and is really very pretty good at it. Very pretty good. Yeah, it's very good at it. But still some stuff gets through. Those are called mutations. I'm not worried about that. Not worried about that specifically. Let's see what we got here. Yeah. Okay. So excision repair, it has to find the damage, so it recognizes the damage, then it excises that damage or cuts it out of the damage region, and then it resynthesizes whatever is needed to refill that region. And in general, it does it very well, and it's very on top of it. So what happens when the repair doesn't work? What happens if we decide we're going to lay out in the sun covered in vegetable oil because we really want that killer tan, and we just lay out there, yes, like my sister did in the 70s, my older sister, and just lays in the sun? Well, those photons are going to hit your DNA, and sometimes it's going to mess them up. And when it messes them up, well, sometimes the proofreading is not going to catch it, and that mutation will go along. And if it's the wrong kind of mutation, if it's a mutation that, say, makes apoptosis stop, then you got something we call cancer. Cancer is generally caused by mutations. Can be caused by some viruses as well, but that's not within our purview right now. So let me see if I can get out of here and back to my lovely visage, which I know you'd rather be looking at, right? Yeah, here we go. I'm back. Yay. Well, that was our first lecture. Yay. And you're probably in your pajamas, right? I'm going to try and put 2 of these up a week, so we would normally have 2 lectures a week. And I will do it pretty much like this because I'm totally an amateur, so bear with me. If you have any questions, absolutely email me. This is my job, right? I know it's easier to talk to me after class, but, well, that's not going to be happening anytime soon, right? Email me. I especially need to know any of you out there who may not have internet access, which, yeah, is logical, right? Because if you don't have internet access, you're not watching this. Well, you get what I mean. I need to know that if you just happen to get somewhere where you can watch this, but you yourself don't have internet access and it's very difficult for you, don't have a computer, let me know. Email me, whatever you need to do. I hope that was at least mildly entertaining. I know it's pretty dry material. Again, there are going to be several videos up in general, lots of it review as well. There will also be videos that I'll put up along with this one that I'll put links up to that relate to this stuff. I've already got some Griffith and Hershey and Chase stuff up. I will be putting up some replication stuff, videos that show it visually that will make it make much more sense. I don't know yet when I'm going to do the exams. Probably going to be next week, maybe the week after, but it's coming up. But I'll let you know, and you will have time in which to do it, right? Okay. So again, any questions, email me. Anything that you would like to see in these videos that I can actually do, let me know. I'll do my best. We're going to do our best to muddle through this semester. I'm going to make it as painless as possible. But again, let me know if there are any issues. Thanks so much, guys. I will have another video up in a couple of days. Thanks so much. Be safe.